we're talking about this morning. I've actually been a ChemAxon user for the last 10 plus years. So, first as a medicinal and bench chemist, and then transitioning into the informatics space. So it's really amazing to see and to be a part of the growth. Not just with the people and the products, but kind of how the whole ChemAxon community has evolved. So I'm going to talk a little bit today about how Tango Therapeutics is using the ChemAxon tools to build out our research informatics platform, and how we're thinking about using these tools and others for early stage drug discovery. So Tango Therapeutics was named one of the Fierce 15 by Fierce Biotech last year. We are a company that's focused on cancer drug discovery and development, focusing on the genetic principle of synthetic lethality. We're applying a unique strategy to advance novel targets in genetically defined patients. So it's really about patient selection, patient populations for our treatments. The core of our research focus really is on developing a product engine that harnesses CRISPR technology for high throughput target discovery and then validation. So again, you'll hear a lot of buzzwords when you come to talks like this and think about how biotechs are launching, and one of them is that it's a data-driven organization. I think that anyone in research and in development is data-driven, but so it's really what are you doing with that data? And we're developing a proprietary analytical pipeline to help elucidate our targets. We've assembled a very experienced and very talented small molecule drug discovery team to really prosecute the more traditional drug discovery components. Kind of piggybacking right off of what Paul said, talking about third rock companies. So Tango is a third rock backed company. We launched a little bit over a year ago. We're based in Cambridge, right around the corner, and currently have 29 full-time employees, a fully operational R&D platform and in lab space, and we are about less than 2 months away from moving into some brand new lab space just around the corner, so we're very excited about that. Since our launch just over a year ago, we have in place our target discovery and validation platforms in which we are screening both public and internal cell lines. We've identified and validated 7 novel targets to date and have 3 drug discovery programs that we're in various stages of running right now. We have an external collaboration team already established and integrated, and so I mentioned we're under 30 full-time employees. We have target discovery and validation platforms running. We have 3 drug discovery programs going. Having this external collaboration team built and sort of scattered throughout the world is very important to enabling us to do the breadth and the volume of work that we have. With that external collaboration team comes the problem of bringing that data in-house and really trying to not just generate data, but really integrate it and make sure that our internal team can make the most of it. Tango's IT infrastructure, again, as Paul mentioned, we're a company that's leveraging the cloud, that's leveraging SaaS deployments for all of our informatics solutions. And I'd go a step further and say that it's not just our informatics, whether it's research, clinical, what have you, but all of our systems that we bring in-house. We're really trying to leverage cloud infrastructure or SaaS deployments where appropriate. I don't have to go into the details of this, but we're leveraging the cloud and SaaS deployments will reduce our IT overhead. It makes the tools easily scalable and highly available. And one of the things that we're really focusing on is limiting the number of thick clients, so moving to as many web-based applications as possible. When we try and think about which solutions to bring in-house, we're balancing a couple of schools of thought. The best in breed, where you go out and you just pick the best product for every use case, versus a single platform where you make some concessions on the strengths of some products, but in general, you've got one streamlined platform. And so we've adopted a bit of a hybrid approach in which we're identifying our critical systems, going with best in breed in those practices, and then for some of our non-critical systems, trying to pepper in a bit of a single platform approach. I like to put this slide up when I talk about how Tango is thinking about our informatics strategy, because really at the end of the day, any of the systems that we bring in place for research informatics, the goal is to aid the DMAT cycle, which is what our scientists and our drug discovery team really uses to iterate through their projects, design, make, test, and then analyze. As you can see, we've balanced a number of cloud and SaaS applications, and then mixed in a number of ChemAxon tools throughout the process. As Aurora mentioned, we currently are using Plexus Design for our enumeration tool, compound registration, and then we have a number of physical, structural, and molecular properties calculators that ChemAxon offers that I think are among the best in the industry. So Plexus Design at Tango, we're using this really to aid our hit-to-lead efforts, which focus on the exploration of new chemical space out of your HTS screens or out of core scaffolds that you may have. Plexus Design offers our chemists to enumerate and then really evaluate both commercially available and sort of proprietary compounds within the context of a scaffold. We've then integrated these plugins that I mentioned, the physical molecular descriptor plugins, into Plexus Design to do further filtering and analysis. We've also taken those plugins and integrated them into some of our data visualization tools that we have, so that way we're carrying forward industry-leading technology into our decision-making. Right now, it's a bit of a manual process to export out our data sets from Plexus and into our data visualization tool. But again, it's very standard and very easy to accomplish. So our team is well suited for what we're doing right now, and we're working closely with the ChemAxon team to kind of provide a bit of roadmap on that. So when you think about critical research systems, I would argue that the ELN is one of the integral parts of any R&D platform. I mentioned again that we have external collaborators, and so one of the things that Tango has decided to do is outsource our chemistry to our CROs. With the integration of our ELN, which is SaaS-based, and our compound registration, which is internal, what we've been able to do is actually allow our CROs to directly upload compound registration data from the ELN. Paul alluded to the best practice setup using an ELB to really set it in a secure and defined manner, and so we've gone through and done that. And again, just by leveraging the ChemAxon native APIs, we can actually have our CROs send compound data directly into a staging area inside of our own VPC for internal QC and then acceptance. The CRO does receive a submission ID so they can track progress. We can continue to communicate back and forth, but they're not exposed to our corporate compound IDs, which we hold internal. One of the real beauties here is that this integration is vendor supported. So, so from our perspective at Tango, there is nothing that we need to maintain here. We're able to really just hammer on this. Another integration point that we've been really really strong in putting in early is compound registration and our results management. So one of the keys to learning anything about the compounds we're studying is to make sure that we have strong control over our results management. And so this allows us to standardize how experimental results are stored, how they're extracted, how we're thinking about them. Consistent data types obviously will allow us to build out a great deal of visualizations and perform sort of basic analyses that if we're spending significant time thinking about cleaning up data, that we're not thinking about doing science. So what we've done, it's actually really simple. We just have that all of our experimental results are validated against the compound registration, and that will ensure that the articles exist, they're properly formatted, that we haven't dropped any of the leading zeros, and that when we report a value, it tracks back to a real number. Again, this was actually a really simple piece to build in, but is critical in making sure that we're maintaining data integrity and data hygiene at an early stage. So just in summary, we have deployed successfully self-hosted instances of Plexus design and compound registration. We've integrated the ChemAxon calculator plugins to our library enumeration deployment, as well as some of our data analysis tools. I left it off of here by mistake, but I think that one of the big wins for Tango, and really thanks to the help of ChemAxon, we've integrated our ELN, our chemistry ELN, with our self-hosted compound registration, and then again the compound registration integrated with management, with our results management. Things that we're looking to do and that we're looking to partner with ChemAxon to push forward would be actually a integration of our document repository to the compound registration. So when we're doing our CROs are doing library-based work, we can actually just take full full SD files and load those in without having to do it in a manual or iterative process. We're constantly looking at ways to pull the utility and the data out of Plexus design and into both our data visualization tools and then into the ELN for streamlined synthesis. And then the acknowledgements. It's a full team. There's plenty of people that have been left off of here, but the team internally at Tango, the backing of the Third Rock platform team, and then the whole ChemAxon team. So I'd like to thank you for your time, and if there are any questions, happy to provide more details.
Have you gotten closer to the validated system stage? Have you tried this development platforms that they're talking about from Amazon? Or are you still pretty early on?
We're still in research stage.
And just a quick scientific question.
Yep.
So you use CRISPR to knock in genes and then see if you get a cancer cell line, if you get unlimited growth? Is that how the technology works?
Yeah, so I think it's a combo, so we'll do it both ways, knock in and knock out genes, and try and find the gene pairs that will that will lead to either cell death or still continue with cell viability. Really making
It's not single gene. You're actually looking at multi-gene combinations.
Correct, and that's the yeah. So that's really the principle behind synthetic lethality, is that it's a mutation or a perturbation in one gene partner makes the entire system more susceptible to targeting the another gene for cell death, whereas if you target just that one partner and there is no mutation or perturbation, you still have cell viability. So it really opens the gap in terms of your therapeutic threshold. And again, that leads back to the patient selection and genetically derived profiling.